I see the world in snapshots, like the frames of a movie, but most of the frames are missing. This was said in 1983 by a German female patient who had experienced a stroke and damaged her middle temporal area in both sides of her brain. She could still perceive colors, forms, and sizes of objects, but saw stilled motion snapshots rather than fluid movement. She had difficulty judging car distance when crossing the street, and often overflowed her teacup from her failure to see the motion of the tea being poured. In addition, she could not perceive facial movements during conversations with others. Although a rare condition, this patient was diagnosed with visual motion blindness. Visual motion blindness is defined as a selective disturbance of visual motion perception, despite intact perception of other features of the visual scene. Scientifically, it's termed cerebral echinotopsia and is often caused by cortical lesions or damage outside of the striate cortex in the middle temporal area, referred to as MT or V5. To better understand motion blindness, we need to understand its foundations in the visual system. The area of the cortex just beyond the primary and secondary visual areas, V1 and V2, was originally referred to as the sensory association cortex. It was unexplored and considered mismatched throughout a major part of visual research. In this region, vision was believed to be recognized and associated with other senses. In 1971, however, Samir Zeki discovered the visual mapping system of this region. As research progressed, this brain region was renamed the middle temporal cortex. Zeki established that the MT was made up of specialized visual maps, each containing distinct representations of the visual field. Cells in this region did not respond to color or form. However, an astonishing 90% of these cells did respond to movement in a particular direction. Further research showed similarities between these cells and other cells in the visual pathway. Research has shown that cells in the MT directly connect to the layer of direction-selective cells in the primary visual cortex. These cells in the V1 are organized in columns by their orientation selectivity. But research has shown that MT cells share this similar architecture. These cells are organized in columns by their receptive orientation, but are also arranged according to their preferred direction of movement. Most research on the MT continues to focus on studying the cells and their response to movement. Current research has shown that goal-directed behavior depends on the ability of the visual system to code object motion and position. Most of these studies measure what is termed manual following response, which is defined as the deviation in trajectory of reaching movements in the presence of background motion. One specific study that we will be discussing was done by Professor David Whitney at the University of California. Whitney took 7 subjects and divided them into 3 subject groups. In the experimental group, the MT was stimulated using TMS. The sham group heard the stimulation but did not receive it, while the control group received TMS stimulation in a brain region unrelated to manual following response. The subject groups were then adapted to visual motion shown by the image on the left-hand side of the figure. A light was moved in a single direction across the region shown by the alternating gray and white box, while the subject was told to stare at the bullseye. The participants were then subjected to a test phase, where they were flashed a target light in the motion-adapted region and told to point to its placement on the screen. This procedure was repeated for 120 trials. During this study, Whitney attached infrared markers to the index finger, recording the finger's trajectory and landing point, and measured the error of the subject's reach. The graph of his results shows that the MT stimulation decreased subject error and illustrates the impact of the MT on processing visual motion. In Whitney's second experiment, he used the same general setup as before, but added an additional reference target that was flashed below the motion-adapted region. The subjects were then asked to judge the alignment of the flashes. His results are shown in the presented graph. The white bars represent subjects that received MT stimulation, while the black and gray bars indicate the sham and control groups respectively. Both the sham and control groups perceive the 2 bars to be misaligned following motion adaptation, but the MT-stimulated experimental group showed a decrease in error in alignment perception. As research like Whitney's on MT cells accumulates, we can better understand the significance of the middle temporal area of the brain in visual motion perception. Hopefully, we may better treat patients like the German female and provide them with a greater ability to live a more functional lifestyle.